All right, so let's do a recap of what we did in our last lesson. We started to look at the structure of hemoglobin, and then we also looked at the function of hemoglobin and some hemoglobinopathies or diseases that are associated with the structure of the protein part of hemoglobin. So we talked about the structure of hemoglobin being a tetrameric compound consisting of 4 protein subunits. And these 4 protein subunits, each of them is having the prosthetic parts, that is the non-protein parts being the heme. We saw the function of hemoglobin being the carrier of oxygen from the lungs to the tissues for cellular respiration, and then the transport of carbon dioxide, which is a byproduct of cellular respiration, to the lungs to be exhaled. Then we actually saw that hemoglobin could transport that byproduct carbon dioxide, just 15% of it, back to the lungs for exhalation. And the rest of the 85%, we saw the mechanism of its elimination from the body being the formation of carbonic acid through the combination of carbon dioxide with water, that reaction which is catalyzed by carbonic anhydrase, and then the dissociation of the carbonic acid into protons, or call it hydrogen ions, and bicarbonate ion. And the bicarbonate ion gets transported freely in the blood, whilst in the blood to the lungs, whilst you have the hydrogen ions binding to the hemoglobin molecule, transported also to the lungs, where the hydrogen ions have a reverse reaction with the bicarbonate ion to form the carbonic acid back, and that carbonic acid gets dissociated into carbon dioxide and water and gets exhaled as the carbon dioxide gas and the water vapor. So we saw that function of hemoglobin compared to myoglobin, which is a monomeric compound, meaning it's having only one globin and one heme attached to it, or one heme as its prosthetic part, and myoglobin playing that intermediary role in the transfer of oxygen from the hemoglobin molecule when it reaches the tissues. And that process is to facilitate the transfer of oxygen from hemoglobin to the tissues. And this you find myoglobin doing much in skeletal tissues, the skeletal muscle tissues, and cardiac tissues. Then we went ahead and looked at the hemoglobinopathies, or abnormalities that occur with the structure of the proteins of these 4 different subunits of hemoglobin. We look specifically at the sickle cell, and then we looked at abnormalities relating to the alpha globins, being alpha thalassemias, and then we saw also with the beta globins being the beta thalassemias. So generally, we That's what we looked at, these hemoglobinopathies related to the protein part of hemoglobin. Now today, we are going to look at abnormalities that are related to the non-protein part of hemoglobin, and that is the heme. So first of all, we want to remind ourselves of the structure of hemoglobin. Now, we should understand that hemoglobin belongs to a class of compounds referred to as porphyrins. So porphyrins are these cyclic structures made up of 4 different chiral rings. And what I mean by that is that the ring which you see on the right-hand side is a chiral. So this is a heterocyclic compound. So you have this nitrogen on the first position with 4 carbon atoms forming a cyclic structure. This cyclic structure, this heterocyclic structure is a chiral ring. It's a chiral. This compound is a chiral. And you see that in the structure of porphyrins, you have 4 of these chiral rings that are linked together. So you see this heterocyclic ring that is labeled 1 or I. 1, you have 2, also a heterocyclic ring. So you can see the nitrogen and then the 4 carbons forming that cyclic compound. And then here also you see the nitrogen with the 4 carbon atoms forming the chiral ring. You see that for the 3rd chiral ring and then the 4th chiral ring. So these are 4 chiral rings, and they are linked together by what is called the methynyl bridges. So you see you have the link between 1 and 2. That is a methynyl bridge. And you see the carbon which is between the chiral ring 1 and chiral ring 2 being referred to as an alpha carbon. And then the second methynyl bridge, that carbon is being referred to as beta. And you have the 3rd methynyl bridge, that is the gamma carbon. And you have the 4th methynyl bridge, and the carbon there is the delta carbon. So you see this general structure of porphyrins. And what you should remember is the porphyrins are made up of 4 chiral rings that are linked together by methynyl bridges. So we move on from here and see some specific examples of porphyrins. Now, don't get intimidated by these structures, as we will take time and look at it step by step. Some of these you would have actually have heard some of these compounds, like vitamin B12. Yes, you have heard of vitamin B12 over and over, but perhaps you may not have thought of it as a porphyrin. But today you are going to see it as such, that it also belongs to those compounds that are referred to as porphyrins. So the structure on the left-hand side is that of vitamin B12, and is also made up of 4 chiral rings. So you see, this is one of the chiral rings. So you see the nitrogen, and then you see the 4 carbons that are bonded on it to have that cyclic structure. So this first ring, this ring that you see here, what I am circling with a cursor, that is a chiral ring. You see another chiral ring here. Here is another chiral ring. So that is chiral ring. You can say the second parallel ring. And you can see a third parallel ring also here. That is a third parallel ring. And then you have a fourth parallel ring also here. That's a fourth parallel ring. And you see all these rings are linked together by methanol bridges. Then at the center of this parallel ring, you have the metal ion cobalt. This is cobalt ion that you see having some interaction with the nitrogen atoms that are found in these parallel rings. Of course, you have some side chains on these parallel rings. So these side chains on the parallel rings will be different in different compounds, in different porphyrins. And that will distinguish the porphyrins one from the other, apart from the metal ion that you find at the center. So you see, because you find that these porphyrins are always having a central metal ion, and so they are being referred to as metalloporphyrins. So that the metal ions at the center actually render the molecule, these compounds, active. Then we see that vitamin B12, we see is being referred to as methylcobalamin. So you see the name, the cobalt ion, which is found at the center of this compound, vitamin B12. The metal which is being found at the center is cobalt. And so you see the other scientific name of vitamin B12 is methylcobalamin. Then we see another porphyrin that is having the central metal being nickel. And this is the compound which is a cofactor of an enzyme in archaebacteria that is responsible for the process of methanogenesis, that is the synthesis of methane from organic matter. So these bacteria, which is actually being applied in industrial biochemistry in biodigesters for the synthesis of what we call biogas. And the enzyme which is reductase, uses this compound, this porphyrin, as a cofactor. And that is why it's being referred to as cofactor F430. And you see that it's also a porphyrin. Is having, this is one parallel ring. This is a second parallel ring. This is a third parallel ring. And then you can have a fourth parallel ring. So you see the four parallel rings that are having that interaction with the nickel ion at the center of the molecule. Now, you see another common molecule, which is also a metalloporphyrin, being chlorophyll. So this is the structure of chlorophyll. Of course, we have different conformations of chlorophyll, or we have different forms of chlorophyll. You can have chlorophyll A, chlorophyll B, C, D, etc. Even C, you have C1, C2, all of that. But I think this is the chlorophyll which is mostly found in plants, which is chlorophyll B. And you find that there is a side chain here, which is more of, I think this is a lipid. This is a fatty acid, which is esterified to one of the parallel rings. So you see, at the center of chlorophyll is the metal magnesium. So even though we have different types of chlorophyll, you find that the metal which is at the center of the chlorophyll molecules is usually magnesium. magnesium. So you see here one parallel ring 2, parallel ring 3, and then you have parallel ring 4. So here again, you see 4 parallel rings that are linked together by methanol bridges, and you have the nitrogen in these parallel rings having interaction with the magnesium ion at the center of that molecule, a porphyrin or a metalloporphyrin. So you see that similarity in hemoglobin, that is in the heme of hemoglobin. You see such a similarity as we have seen of these metalloporphyrins earlier. So you see, back to the structure of the heme, you see it has 4 parallel rings. Here it's labeled A, B, C, and D. And you have the central metal being the ferrous ion, the Fe2+. That is the central metal ion in the heme of hemoglobin. So in heme, this heme, you see 4 parallel rings, and you see the parallel rings being linked by methanol bridges. And thus, you can confidently say that heme is a metalloporphyrin, just like all the others which we have seen. Now that we have established that, yes, heme is a porphyrin or a metalloporphyrin, and the metal that is found at the center of heme is the ferrous ion, we can move on further to look at the metabolism of heme, because it is going to be based on the metabolism of heme that will come to understand the abnormalities that can be related to the heme. But before then, because we are going to look at some series of reactions or processes, we need to simplify the structure of the porphyrin so that the processes that we are going to look at will not be looking too clumsy in our eyes. And to do that, we are going to use what is referred to as the Hans-Fischer model of porphyrins, or simply referred to as the Fischer model or the Fischer's model of porphyrins. And that simply is the representation of the parallel ring by square brackets. So if you take one parallel ring, you see that parallel ring 1 is being represented by a square bracket. So this parallel ring is being represented here by this square bracket 1. And the second parallel ring is also represented here by the square bracket 2. The third is represented by square bracket 3, and then the fourth is represented by square bracket 4. That is the Fischer's model, or the Fischer model for the representation of porphyrins, to simplify the representation of porphyrins. And to be specific about the metal that is at the center of the porphyrin that is being talked about, for instance, we are talking about heme. Heme has the ferrous ion as the central metal, and so you can write the central metal Fe2+ at the center to specify the metal ion at the center of the metalloporphyrin. All right, so now we are going to start to use the Fischer's model in the series of processes of synthesis or the metabolism of porphyrins to understand how these are related to the abnormalities that occur in relation to heme. All right. Now, the first thing we are going to look at is the biosynthesis of heme. Now, let's bring our mind back to metabolism. Now, when we talk about metabolism, what we mean actually is two processes. So two processes make up metabolism. And these processes are catabolism and anabolism. So catabolism being the breakdown of a big molecule into smaller molecules. And anabolism being the buildup or the synthesis of a bigger molecule from smaller molecules. So generally, metabolism, when we talk about it, it is either we are talking about breakdown, which is catabolism, or we are talking about anabolism, which is the build-up or the synthesis. So in this case, when you see biosynthesis, it only means the anabolic process, the synthesis, the build-up of. So we want to see the synthesis of heme in biological systems, biosynthesis of heme. And we see that there are 5 steps that are listed in that process of synthesis. We are going to look at it in the reaction path, which is on the right, step by step, to see what is happening. Now, you remember in our first class, I mentioned to you that you must be patient with yourself. Sometimes, biochemistry, when you see certain things for the first time, it will be intimidating, but you just have to be calm. Take your time, step by step, and you will understand it. If you don't get it so well the first time, though it can be frustrating, it can be painful, the second time, you begin to get it. The third time, you'll get it well. So please be patient with yourself and don't get frustrated at any point in time. So now we start to look at the first step, which is the formation of delta amino levulinate. All right. Now we see the very first reactants at the beginning. These we have seen before. You see the very first here, the blue compound is what? Succinyl-CoA. And quickly, that name should ring a bell, that you can get succinyl-CoA from the TCA cycle or the Krebs cycle. You did the metabolism of carbohydrates. So you saw the TCA cycle or the Krebs cycle in first year. And in that cycle, one of the intermediates was succinate. And there you see succinyl-CoA from the Krebs cycle coming. Then you have also seen glycine when you were studying proteins. Glycine being the smallest amino acid, the simplest of the amino acids. So these two are two simple compounds, succinyl-CoA and glycine. Now, take note that yes, glycine is in blue color, that is intentionally made, and then you have glycine in red color, so that as we move along the reaction chain, you will see which part of a compound is coming from one of these reactants, either succinyl-CoA or glycine. Now, the first thing you see happening is the conjugation of succinyl-CoA and glycine. And you see the CoA, the coenzyme A portion of succinyl CoA is released, is displaced by the glycine molecule, so that you now have a conjugation of glycine with succinyl CoA as this coenzyme A portion on succinyl CoA is displaced. So you now see that, yes, the alpha carbon in glycine is bonded directly on this carbonyl carbon of succinyl CoA. Now, so that is the first compound, that conjugate, which is formed from succinyl CoA and glycine, being referred to as the alpha amino beta keto adipate. Well, there is no need to be scrambling these names in your mind. What I will draw your attention to is the fact that in these reactions, you have enzymes taking place, enzymes that are catalyzing these reactions at every stage. So you see, the first reaction is being catalyzed by an enzyme, which is shaded green, and that enzyme is the delta aminolevulinate synthase. You have pyridoxal phosphate, which is a coenzyme to that enzyme. Then, now the formation of these delta aminolevulinate. You see from the first conjugate, you have some decarboxylation. Carbon dioxide is given out, and then you now have the structure, the delta aminolevulinate. One carbon is out. And so you now have the structure of the delta aminolevulinate. Now, from there on, look at what is happening. You see two molecules of this, the delta aminolevulinate. So this is one molecule on this left-hand side, and this is another one molecule on the right-hand side, the delta aminolevulinate. Two molecules come together, and the reaction between the two molecules of delta aminolevulinate forms a pyrrole ring. This is the pyrrole ring that further goes on to form the ring structure of the porphyrin. So from succinyl CoA, which is a linear molecule, glycine is also a linear molecule. You have a conjugate, and then this conjugate gives you the delta aminolevulinate, and the two molecules of delta aminolevulinate come together to give you the porphobilinogen, which is having that pyrrole ring, which is a pyrrole. So from here on, you see that four of these pyrrole, the porphobilinogen, will now form the ring structure. So from here, you continue on the right-hand side, where you now form the four pyrrole ring structures coming together, being linked by methynyl bridges. Now you have the ring structure, that cyclic structure being formed. And from there on, you see the representation being the Fisher model. And what is happening, as you notice, is that we are having some rearrangement of the groups that are on the rings. The methyl groups, propionyl groups, you see rearrangement. Here is methyl, here is vinyl, here it was propionyl, it has become vinyl. Here too, it was propionyl, it was propionyl, up here it has become vinyl. So it's just rearrangement of these residues on the pyrrole rings that is happening. Call it some form of isomerization that is occurring with some process of decarboxylation. So you see carbon dioxide is out, carbon dioxide is out, some oxidation. So all of these isomerization reactions occurring just to do rearrangement of the groups that are found, the residues that are found on these spiral rings. And then the very last thing that you see happening is that after you have the porphyrin cyclic structure formed, you now have the ferrous ion being embedded at the center of it. And that is being catalyzed by the enzyme ferrochelatase. Now, these step-by-step processes, as I say, can be sounding so hard, difficult to understand and things like that. But really, what I want you to bring your mind to is the fact that at every stage, all these enzymes that are shaded green are proteins. These enzymes are proteins. From the aminolevulinate synthase, aminolevulinate dehydratase, uroporphyrinogen 1 synthase, uroporphyrinogen 3 co-synthase, and so on and so forth, all the way up to ferrochelatase. All these enzymes are proteins. And what does that mean? If you cast your mind back to what we did with gene expression, where you have the process of synthesis of proteins from the messenger RNA. You have genetic codes that are coding for specific amino acids, which come together to give you a protein. And so we'll come to that. Keep that in mind, that you will be having disorders, genetic disorders, which are related to the synthesis of these proteins, which become the enzymes that are catalyzing these reactions of synthesis of heme. That is the most important thing for you to understand, and not necessarily the step-by-step, synthesis that is happening with heme. Yes, if you understand the step-by-step synthesis, you begin to appreciate the fact that, yes, there can be hemoglobinopathies or abnormalities related to that process of synthesis of the heme because of the proteins, which are the enzymes catalyzing these reactions step by step all the way to the formation of the complete structure of the heme. Right. Now, having said that, we now move on to talk about the regulation of that process of heme synthesis. So again, you remember that when we were looking at gene expression, that is the synthesis of proteins along the line, we looked at regulation of these processes in prokaryotes and eukaryotes. So the same thing we are coming to do here, to look at processes of regulation of heme synthesis, since we are talking about enzymes that are catalyzing these reactions. There must certainly be some form of regulation of these processes. Now, one thing you need to understand is that the synthesis of heme takes place in two places in the body. One is in the liver, and one is in the bone marrow. Now, the synthesis of heme, which occurs in the liver, is not responsible for the heme which you find in hemoglobin. The synthesis of heme in the liver is responsible for the heme which you find in the enzymes that are responsible for the metabolism of drugs, of chemicals. It can be toxin, it can be some chemicals that have some therapeutic activity, what we refer to as medicines. And that process is also what we refer to as biotransformation. So now you see that you have heme, which is being synthesized in the liver, which will get incorporated into the structure of some proteins, which are enzymes responsible for the metabolism of drugs in the liver. So these are hepatic enzymes, enzymes of the liver. Most importantly is what is group of enzymes referred to as cytochrome P450, which are responsible for the metabolism of drugs. How about the synthesis of the heme in the bone marrow? So the synthesis of heme in bone marrow, which is occurring in what is referred to as erythroid cells in the bone marrow, are the ones that are responsible for the heme in hemoglobin. That heme, which is synthesized in the erythroid cells, are those that get embedded in the porphyrin structure to form hemoglobin, which is the carrier of oxygen and carbon dioxide in the blood. Now, having understood that, the regulation of heme synthesis in liver is quite different from the regulation which occurs in the erythroid cells of the bone marrow. So we are going to look at these two, how the regulation occurs. So the first is what is referred to as feedback inhibition. And feedback inhibition simply means, yes, you have a final product, which is heme. And when you have heme in large quantities, you have heme in high concentration in the blood, there will be no need for more synthesis of heme. Because of that concentration, you have heme that is giving a signal, is inhibiting the very first process of the formation of the first conjugate from succinyl-CoA and glycine. So heme can act as an inhibitor to the enzyme aminolevulinate synthase. That is a feedback inhibition because you have it in high concentrations. After each synthesis, you have enough of it, and then it begins to give the signal, become an inhibitor to this first enzyme, so that the reaction, the first reaction, does not proceed for the formation of parallel rings, which will then now bind together by methanol bridges to even form that cyclic structure for even ion to come to get embedded into it to form the full structure of the heme. Or you can have what is referred to as repression. So repression too we saw, where you saw when we were looking at the operon concept. You remember that we saw a repressor which was bound on the gene which is responsible for the synthesis of the enzymes that are responsible for the metabolism of lactose. We saw that with lac operon. So the same thing which is happening where you have heme acting as a repressor of delta aminolevulinate synthase. So in that case, it means it goes to also, it's a form of inhibition of the enzyme. So it goes to block, say, the synthesis of this enzyme. Or you can have the inhibition of the transport of the enzyme, the delta aminolevulinase, this first enzyme here, from the cytoplasm to mitochondria. This synthesis occurs in the mitochondria, but in order for that process to occur in the mitochondria, there must be the presence of this enzyme to catalyze the first two reactions for the enzyme, for the process to proceed. And the enzyme is present in the cytoplasm, moves into the mitochondria before you have the process starting. And heme can inhibit that process of transport of the enzyme into the mitochondria. Now, take note that certain drugs can influence the activity of this enzyme, the delta aminolevulinase synthase. Of course, I mentioned that the synthesis of heme in the liver, the heme which is synthesized there, the liver, gets incorporated in enzymes that are responsible for the metabolism of drugs. And what it means is that in the presence of these drugs, when there is the need for their metabolism, there will be an increase of synthesis of heme. The same way, there will be an increase of synthesis of the protein part of the enzymes that are responsible for the metabolism of these drugs. So what it means is that the drugs can act as inducers of the process of synthesis of the enzyme. So you keep that in mind, that drugs can have influence on the activity of these enzymes. Not only medicines, but there can be influence of some chemicals like some pesticides, some herbicides. These toxic chemicals, when they get inhaled into the system, they also undergo that biotransformation, that breakdown in the liver. And so in their elevated amounts in the blood, they can also cause the influence of the activity of delta aminolevulinase in the liver. All right, now we move on to look at the regulation of heme synthesis in the erythroid cells. That is the synthesis which is occurring in the bone marrow. And this is what is specifically responsible for the synthesis of the hemoglobin, because it is from here that you get the heme that we find in hemoglobin. And here we see that the mechanism of regulation is either occurring with uroporphyrinogen synthase or with ferrochelatase. And you see uroporphyrinogen synthase being the enzyme which is responsible for the formation of the porphyrin ring structure. So when you have the pyrrole ring formed, the enzyme which is responsible for bringing 4 of these pyrrole rings, so 4 PBG molecules, 4 of these pyrrole rings, you have the enzyme uroporphyrinogen being the enzyme responsible for bringing these 4 rings together to form the ring structure of the porphyrin. And the other enzyme, ferrochelatase, is what is responsible for the embedding the ferrous ion at the center of the porphyrin structure, which has formed to form now the complete structure of heme. And you have the regulation of these enzymes, the regulation of these 2 enzymes being responsible for the regulation of the synthesis of heme in the erythroid cells. Then it can be also by cellular ion uptake. So it's like you need iron for the formation of this. Even if the iron is present, and this enzyme ferrochelatase is inhibited, so what it means is heme, which is the final product, inhibits ferrochelatase here in this the first form of regulation, where we have heme being an inhibitor to this ferrochelatase, or heme being an inhibitor to this uroporphyrinogen synthase, which is responsible for the formation of the porphyrin structure. It is either you have that heme inhibiting these two processes, or you have the inhibition of the cellular uptake of iron. So if you don't have more iron that is entering the cell, then of course you don't have enough of these ferrous ions present to be embedded in the structure to give you the complete structure of the heme. So these are the forms of regulation of heme synthesis which you find in the bone marrow. Now again, you should also take note that heme can stimulate the synthesis of globins. So the globins that heme will get embedded in to form the hemoglobin structure, those globins are proteins, and heme acts as an inducer of the synthesis of those globins. Because if you have more heme being synthesized, there should be globins present for them to get embedded in to form the full structure of hemoglobin. So once you have more of these heme being synthesized, they become inducers of the synthesis of the globins. All right. Now that we have seen the synthesis, or call it anabolism, of heme in the body, we have seen it both in the liver and in the erythroid cells of the bone marrow. We can now talk about the abnormalities that can occur as far as the synthesis of heme is concerned. Now, remember, we talked about the fact that the synthesis, the very final synthesis of heme, occurs in the mitochondria. So you see here all the reactions that are occurring in this site, which is, you have it in the structure of a mitochondria to show that this is inside the mitochondria. All the reactions occurring here are in the mitochondria, and outside here are all occurring in the cytoplasm, or say the cytosol. All right, having understood that, what I want you to put your mind to is these places which are marked red: 1, 2, 3, 4, 5, and 6. And you notice that, as I mentioned earlier, that you should pay attention to the fact that all these reactions of synthesis are being catalyzed by enzymes. And these enzymes are proteins. And so you can connect it back to what we did earlier on gene expression. Now, the abnormalities that are associated with the deficiency of the synthesis of these proteins, which are the enzymes, are what is referred to as porphyrias. So porphyrias are those defects that you find with the synthesis of heme. The defects, the abnormalities that occur, which are associated with the synthesis of heme. So you see, in the first place, this is 1, and that 1 is also represented below here. 1 is referred to as what? Acute intermittent porphyria. And that is related to a deficiency in the synthesis of the enzyme uroporphyrinogen 1 synthase. If you have a deficiency of uroporphyrinogen 1 synthase, what it means is that the compound porphobilinogen, which it is converting to give you the next compound, which is uroporphyrinogen 3. These compound up here, because you are having a deficiency of this enzyme, you have an accumulation of this intermediary compound, which is porphobilinogen. What it means is, once you are having the accumulation of this in the blood, well, you can have all of these now going through the path, this other path, an alternative path, giving you what? Uroporphyrinogen. And if you have uroporphyrinogen, this, as you can see, is photosensitive, like it reacts with light to give you uroporphyrin 1. And so you will see that for such persons, they are having some photosensitivity. In some cases, maybe if you have a lot of these porphobilinogen, which is being accumulated in blood and being excreted through urine, for example, upon exposure to oxygen and light, you have this compound, the last compound, uroporphyrin, forming in large quantities, and it will have urine having a dark color, for instance. A similar thing is happening also with the second enzyme, which is also playing a role in the conversion of this porphobilinogen to the next compound, which is uroporphyrinogen. It is the same thing, similar thing, which is occurring here too. If you have this enzyme, uroporphyrinogen decarboxylase, this is number 3. If you have this also being in deficiency of this, so deficiency means you are having, less synthesis of the enzyme, that is the proteins that are responsible for the conversion of this uroporphyrinogen 3. Then you see that this compound is also light sensitive, it's photosensitive. It will also react and give you uroporphyrin 3. That will also cause some photosensitivity of the individual. You go on, here too, you see that coporphyrinogen oxidase, if there is a deficiency of it, a deficiency in the synthesis of that enzyme, and then you have an accumulation of this coproporphyrinogen 3. This will also take an alternative path where it is also going to react with light to give you proporphyrin 3. So there is that photosensitivity also occurring where you are having some pigmentation of urine turning dark color and other things. Maybe the skin can be reacting to some of these things. Now you see, if you have the 4th one, this is the 5th one. If it is protoporphyrinogen oxidase, if you have it, if you are having a deficiency of that, you are going to have a porphyria, which is being referred to as variegate porphyria. The last one is the 6th one, the enzyme ferrochelatase, which is responsible for incorporating the ferrous ion in the porphyrin structure to give you the complete structure of heme, which will then bind to the globins to give you the full structure of hemoglobin. If you have this also, being in deficiency, then you are having a porphyria, which is being referred to as protoporphyria. We are going to look further at the symptoms that are related to these porphyrias. And these porphyrias are divided into two. Like we said earlier, that the synthesis of the heme is either happening in the liver or it is happening in the erythroid cells of the bone marrow. So the porphyrias are divided into two. Hepatic. So the hepatic are 1, 2, 3, 4. These are occurring in the liver. Whilst you are having erythropoietic, these are happening in the erythroid cells of the bone marrow. So that if you have what we saw earlier happening, and then the enzyme defects, the enzymes, the deficiency of which cause these types of porphyrias, and then you see that there are some symptoms that are written, like the abdominal pain for, say, acute intermittent porphyria. We mentioned much about photosensitivity, and you see it occurring, photosensitivity, abdominal pain again. You see photosensitivity, increased hemolysis, breakdown of red blood cells occurring. Then you also see one interesting thing, which is what? Neuropsychiatric symptoms, which are also common in these instances. So that what happens is, when you have accumulation of some of these compounds that are not being able to get converted to the next stage for the synthesis of heme, these compounds become toxic, and some of them can actually break through the blood-brain barrier and cause such toxicity of the nervous system, and that can result into some neuropsychiatric disorders. And some of these neuropsychiatric disorders are very rare, but they cause very severe psychotic diseases. Some can be some very high level of depression, some delusional behaviors. So one of the common delusional behaviors that are with these rare neuropsychiatric disorders is related to, you see, those of you who watch movies, and you see some of these movies are depicting human beings that grow long teeth and they suck human blood. They refer to some of these movies as vampire movies and things like that. Some of those things are actually related to these porphyrias, which in some European folk tales, European stories, have human beings that turn into such wild animals. So it's some form of a delusional condition of these people, that they begin to behave as animals. And a very common example that you find being given a lot of times in neuropsychiatry is the story of Nebuchadnezzar in the Bible that turned into a lion. You know, that actually, looking at it scientifically, in some books, you see it being explained as a form of these porphyrias that is, you call it werewolf, or you refer to it as a lycanthropy or lycanthropic disorder, or some they refer to it as boanthropy. So these people actually have some form of hallucination, and they begin to see themselves as some animals. Boanthropy, the person will begin to see itself as some herbivore, like a cow, an animal, and begin to eat grass and things like that. And lycanthropic disorder, the person sees itself as some wild animal and begin to put up very wild behaviors, things like that. So you can see some of these very rare conditions occurring with these porphyrias. Now we move on, and then we look at degradation. So what we looked at earlier was synthesis, which was anabolism. Now we are looking at degradation, which is catabolism, the breakdown. So we see here the hemoglobin molecule. So this is a hemoglobin molecule. We have seen this before. The heme is represented at the center with the globins, which are alpha globins, beta globins, these adult hemoglobin. Now, the breakdown of this hemoglobin will give you the globin chains. These are proteins. So the beta globins break into their separate subunits. The alpha globins also break into their separate subunits. And they can further get broken down into amino acids. They are proteins, so they are made up of amino acids. And these amino acids can undergo further breakdown, say deamination, the amine group is removed, and then the carbon chain can go into the synthesis of other molecules, maybe in the metabolism of carbohydrates or metabolism of lipids. You can have the carbon chain going there. Or these amino acids can actually be reutilized in the synthesis of proteins in the body. Then now we come to our main thing, which is the heme. After you have gotten the globin chains being broken down, you are now left with the heme. So what happens to the heme? So first of all, you see the heme has this ring structure of parallel rings, as we saw earlier. The first thing which happens is that you have this cyclic structure broken into a linear structure. So heme is broken into biliverdin, and biliverdin is a linear structure. Yes, the 4 parallel rings are there, all right, but these 4 parallel rings are not forming a cyclic structure. And the ion, the ferrous ion at the center, is released and gets oxidized. So that's an enzyme, heme oxygenase, gets oxidized. And then you are having some sides of the chain getting oxidized. These nitrogens in these terminal parallel rings are getting reduced with hydrogen ions. And further, you have biliverdin reductase, the enzyme, which now converts this. What it does is you have further reduction of biliverdin to form bilirubin. Now, in some organisms like reptiles and birds, yes, that breakdown up to biliverdin. Biliverdin becomes the last, the very final product, and biliverdin can be excreted directly in those animals. But in humans, the reaction continues further to form bilirubin. And bilirubin now binds with blood albumin. So you have a complex of bilirubin and albumin, which then moves into the liver. In the liver, you have the process of conjugation with glucuronic acid. So you see glucuronic acid binding on these 2 parallel rings at the center of the linear structure. And that conjugation reaction is responsible for making the compound more soluble to be transported. Now, you see that because you see these parallel rings are not, they are not soluble in the aqueous environment, but the attachment of glucuronic acid makes it now soluble, and they can further get broken down to urobilinogen, coming to urobilin, and then they get excreted. So from the kidney, you have urobilin, and then you have it being excreted in urine. So urobilin will be responsible for the yellow color of urine. Or you have from here, urobilinogen, and then you have the formation of stercobilin that will come out in stool or in fecal matter. And you see this reaction which is occurring here being represented more simply here, that from blood where you have bilirubin albumin complex that moves to the liver, and in the liver you are having the conjugation with glucuronic acid to form that conjugate of bilirubin glucuronide, which then now gets excreted in urine. And I mentioned earlier that yes, that conjugation reaction is responsible for making bilirubin now soluble in aqueous environment, so that it can easily be excreted. So it will be excreted through the bile ducts and then to the kidneys, and then further conversion, and then it gets excreted to urine, or it can get excreted out of the body through fecal matter. So that is about the degradation of bile pigments. Now, what can be associated with these process? So you can have what is referred to as jaundice, so various forms of jaundices. And it can be some hemolytic jaundice, which is caused by increased hemolysis of erythrocytes. So what it means is that if you are having an increased level of breakdown of red blood cells, and you are having a lot of bilirubin being produced, these are pigments, these are bile pigments that can get accumulated in the blood and can cause some pigmentation. So you see the pigmentation of the eyes being yellow. You can see pigmentation of the palm being yellow. Can be hepatic jaundice, which is caused by a dysfunction of the liver. Like we saw there, that yes, this process of breakdown of heme is occurring in the liver. And so if you are having a dysfunction of the liver, you will be having a dysfunction of this process of the breakdown of the heme to be excreted. And so you'll be having an accumulation of these bile pigments, which will be increasing the pigmentation of urine. It will be increasing the pigmentation of fecal matter. Then you can also see that, yes, in the liver, you have the bile duct, which transports these products of the breakdown of heme, the bile pigments, to bile. So if you are having the accumulation of bilirubin, for instance, and I mentioned that bilirubin is not soluble and has to get conjugated to glucuronic acid to make it soluble. So if you have it in high quantities and they go to be blocking the bile duct, which is responsible for the transport of these bile pigments and then some bile salts from the liver to the bile, and then they can get excreted through feces. You have the obstruction of the bile duct, which is now referred to as obstructive jaundice. We can also have, say, in babies, some of these forms of jaundice occurring due to the fact that as babies, when they have not yet developed fully liver function, and you have an accumulation of these bile pigments, you can be having some form of some form of jaundice due to the accumulation of these bile pigments. Some of these are some syndromes like Gilbert syndromes. All of these are related to the fact that it is either you have some deficiency, deficiency in the enzymes that are responsible. So here too, you see that there are some enzymes, reductase, some bilirubin glucuronyl transferase, which is responsible for that conjugation reaction specifically. So when you are having a deficiency of these enzymes, then of course you will be having a defect in the conversion of bilirubin to its conjugate to make it more soluble so that it can be excreted. And thus we'll be having the what we call the accumulation of these bile pigments, which will be resulting into those yellow pigmentation of the tissues. That it will be shown in the eyes, to be showing in the palm, and you can see it's some of these being excreted directly. Urine is very yellow. You are having fecal matter getting dark because of a lot of these bile pigments being excreted directly. So you see some of these forms of jaundice also occurring, which is related to the abnormalities with the process of degradation or catabolism of heme. All right, so we end it there. Now we can have some questions, some contributions, and then we can close. So the floor will now be opened. So if you want to ask any question, you can now unmute your mic, or you want to say something, you want to make a contribution, you can put on your mic and then you can talk. So if you have anything to say, you can put up your hand, I'll call you and then you speak. So the floor is open now.
Yes, sir.
Yes, Joseph Bills.
Yes, sir. I want to find out.
Joseph.
Yes, sir.
I can't hear you.
Hello, sir. Can you hear me, please?
Yes, I can hear you now.
Okay, I wanted to find out, aside the deficiency of the enzymes, is there any other thing that could affect the enzymes? Because you say the deficiency, as in the insufficiency of the enzyme or lack of the enzymes, would be one of the reasons for this problem. Aside the deficiency, is there any other thing that could affect the enzyme, which will result in some of these challenges?
Okay, apart from deficiency, what can happen will be that though the enzyme is there, it will be inhibited by some chemical, can be some drug which is being taken, and that drug tends to be an inhibitor of a particular enzyme. So apart from deficiency, there can also be inhibition. The enzyme is there, but there is inhibition of it by a chemical which has been introduced in the body. Yes, any other?
Sir, please, can you help us to know the difference between D-bill and T-bill?
The difference between what?
D-bill and T-bill.
What is that?
The D-bill,
I don't know.
In
Yes.
direct bilirubin and then the total bilirubin. I have heard that in a test. Test for renal function test. Sorry, renal function test. That's kidney function test. I've heard of D-bill and T-bill.
Well, I don't know the difference. Total bilirubin and direct bilirubin.
bilirubin, yes, sir.
Yes, if I say I know, I don't know. I don't know what the difference is. Yes, any other? So when we go, we can go and read around it and see what it is. Total bilirubin and direct bilirubin. What the difference is really about these things.
Sir, please, one more thing.
Yes.
Does UV light have any effect over bilirubin formation?
Well, I get your question. You know, in the occurrence of jaundice, for instance, we have what is referred to as phototherapy. So that when you have the accumulation of some of these compounds like bilirubin, porphyrinogen, and things like that, when you are exposed to light, because we saw earlier that some of those intermediates. So let's go back to those places and see. So we see that some of these intermediates are light sensitive. So that if you have an accumulation of porphobilinogen, for instance, if you have an exposure to light, then you can have the conversion of this compound into uroporphyrin to be excreted in urine. So that, you know, that conversion is happening in the body before you are having the excretion of it. So yes, it may not be directly the bilirubin itself that is having that reaction with light, but the specific intermediates that are light sensitive. It is those ones that get reacted with light to form the final products along those reaction chains for them to be excreted out of the body. So that is about the phototherapy in terms of jaundice.
Okay, sir.
Yeah.
Yes.
Can some of these deficiencies of the enzymes?
I can't hear you well.
The deficiency of these enzymes, can they be corrected?
Well, some of these deficiencies are corrected by some blood transfusions. Some of it can be corrected if it is not congenital, because some of these are congenital from birth, they are hereditary. But if they are acquired, that is, if it occurs because of the presence of some drug, some chemical, then of course there can be reversal of them with some other chemical compounds as drugs, which go in to activate these enzymes. So yes, they can be corrected, especially when they are acquired, not congenital. They can be corrected. Yes. No other. So we can go and learn more. Today we are only 25. Okay. I see nobody talking again. So I guess we can say good night to ourselves till the next time we meet. Right?
Yes.
Yes, sir.
All right.
Yes, sir.
So
Good night, sir.
Good night to all of you.
Thank you.
When you sleep, dream of biochemistry well.
Okay.
No problem, sir.
All right. Bye-bye.
Bye.
Bye.
Chemistry well. Hey, Charlie.